In the last session, we started to see questions from level 2. So now in this session, we will continue few more questions from level 2 and then continue to level 3 questions. So now here we have a question from level 2. The membranous area between the cranial bones of a fetal skull are called. Here, the bones in the cranium or the cranial bones of the fetus, they are not fused. So are not fused. So let's see the options. The first one, sutures. So these sutures are immovable joints. The sutures are immovable joints which are present between cranial bones. Present between cranial bones. Right. The next one coming to areolas. So here the meaning of areolas is it's a pigmented area. It's a pigmented area present around nipple of the mammary gland. So it is present around the nipple of the mammary gland. The areola is a pigmented area. Now coming to foramina. So here this foramina is any opening. So foramina is any opening that allows nerves, veins, arteries to connect from one part to another. So they are called foraminas. And coming to these fontanelles. So these fontanelles are membranous gaps which later will become sutures. So in the beginning, I mean when the baby is born, the skull bones are not fused. So they later get ossified and then they finally get fused and become immovable joints, what we call them sutures. So the membranous area between the cranial bones of the fetal skull, they are called fontanelles. So the option C is right. So the next question, pick the correct order of events during contractions. So the correct order of events during contractions. So let's see here, we know the sarcolemma that invaginates, okay, into the sarcoplasm, and you will see the presence of the sarcoplasmic reticulum. So the sarcoplasmic reticulum present on either side of this invagination. So here, okay, so this is what I call it T tubule. That stands, the T stands for transverse tubule. And okay, on either side of this T tubule we have is the sarcoplasmic reticulum. So this sarcoplasmic reticulum is storehouse of calcium, okay. So here from the neuromuscular junction, so when the information is passed, so the information will, okay, pass through the sarcolemma into the T tubule and finally reaches the sarcoplasmic reticulum, and from the sarcoplasmic reticulum there will be release of calcium ions, and these calcium ions will later bind to the troponin, and that initiates muscle contraction. Okay, so this is the sequence of events that take place during the muscle contraction. So now let us see the statements. The first statement: acetylcholine is secreted by neurotransmitters uh, okay, present in calcium storing bags. No, that is a wrong statement. So acetylcholine is released from the synaptic knob. So here is the synaptic knob that makes a neuromuscular junction with the sarcolemma. So here acetylcholine is secreted by neurotransmitters present in calcium storage bags. That's a wrong statement. Calcium ions are released, then stimulate the T system. So that is okay. This T tubule with the sarcoplasmic reticulum on either side, that they together form okay a triad system that we have seen in our previous sessions. Okay, so then muscle contraction. So this statement is not a correct statement. Acetylcholine is secreted in the synaptic junction. Okay. Calcium ions are released from myosin. No, calcium ions are not released from myosin. They are released from sarcoplasmic reticulum. Then the T system excited muscle contraction. So this is also a wrong statement. Acetylcholine is secreted in deep sarcoplasm. No, the acetylcholine is a neurotransmitter that is released. So here is the neurotransmitter that is released, and this is the acetylcholine. It is released from the okay end of the neuron. This is wrong. Through T system, stimulus spreads into the sarcolemma, of course, and calcium ions are liberated. Okay, so but this is not again a correct statement. Acetylcholine is secreted in the synaptic union. Yes. Through T system. Yes. The next comes is the T system stimulus spreads deep into the sarcoplasm. Okay, that reaches finally the sarcoplasmic reticulum, and the calcium ions are liberated. Yes, and then the muscle contraction begins. So the option D is the right option. So the next question: vertebro-arterial canal is present in. So here the vertebro-arterial canals uh, it is present in. So let's see which of these options is the right. So here there are vertebral arteries. So these vertebral arteries arise from subclavian arteries. And here the vertebrae in the cervical region, in cervical region we know the cervical region is in the neck region. So in the okay cervical region they contain openings, okay, small openings. They contain okay minute openings, right, in each vertebral body, in each vertebral body, and through these openings the artery enters, okay, and carries blood to brainstem. Then it carries okay to spinal cord, then to cerebellum, and to the posterior region of brain. So it supplies blood to all parts of, okay, to all these parts. And the opening is what I call it the transverse foramen. It's called transverse foramen, through which the artery enters. Now, here these vertebro-arterial canals are present in the, as I said here, in the cervical region. So that is in the cervical vertebrae. So cervical vertebrae are the vertebrae present in the neck region, whereas the thoracic vertebrae are present in thoracic region, and the lumbar vertebrae, they are present in the lumbar region. Okay, so sacrum we know, okay, below the lumbar region it is present. So vertebro arterial canal is present in the cervical vertebrae. So option D is the right. The next question: select the correct statement regarding the specific disorder of muscular or skeletal system. So we need to find out the correct statement here. So first one is muscular dystrophy. So this muscular dystrophy is progressive degeneration of skeletal muscle, and that is due to genetic disorder. We know it. So here age related shortening of muscles is not matching. The next one, myasthenia gravis, is a autoimmune disorder. Yes, it is an autoimmune disorder which inhibits sliding of myosin filaments is not correct. Right, so actually it destroys the acetylcholine or the neurotransmitter receptors with which, okay, the transmission of impulse doesn't pass to muscles. So myasthenia gravis is an autoimmune disorder, that is right, but it is not the one that plays a role in inhibiting sliding of myosin filaments. So this is again a wrong option. Gout, so we know here the gout is due to accumulation, it is caused due to accumulation of uric acid crystals. So it is caused due to accumulation of uric acid crystals that causes inflammation of joints, causes inflammation of joints. Okay, so here gout, inflammation of joints, yes, but not due to deposition of calcium. So hence this statement is also a wrong statement. So coming to osteoporosis, okay, decrease in bone mass, yes, and higher. So as the bone mass decreases, there is more chance of fractures that advance it with the age. So this is also a right statement. So the option D will be the right option. So the next question, name the bone which is not attached to any other bone. So here when we see the sacrum, so this sacrum is part of vertebral column, and this one anteriorly it is attached to lumbar vertebrae, and posteriorly we see it attached to coccyx. So this one is in connection with bone. Then coming to the femur, so here in case of okay, the femur, it's a part of hind limb, and this one articulates with the pelvic girdle, and also with the tibia and fibula. Now coming to these tarsus, so these tarsus are, we call them ankle bones, and these ankle bones are seven in number. So you can see in this picture, so here it is, okay, 1, so this is 1, this is 2, 3, 4, okay, I mean 5, right, and then 6, and this is 7. So you have total 7 ankle bones. And coming to this hyoid bone, so this hyoid bone is a U-shaped bone, and this one is not connected to any other bone, and present below the buccal uh, cavity region. So it is present below the buccal cavity region, supporting the larynx, tongue, etc. And this bone is not in connection with any other bone. So answer is D. Hyoid bone is the bone that is not attached to any other bone. That's all the questions from the level 2. Now let's see the questions from level 3. So the next question, true joints are. So here coming to true joints are the joints that are characterized by the presence of fluid filled cavities, characterized by fluid, okay, synovial cavity. So that is characterized by fluid filled synovial cavity. So here coming to these synchondroses, so these synchondroses are cartilaginous joints. So these are cartilaginous joints. Okay, so they don't have the fluid filled synovial cavity. Okay, then coming to syndesmoses. So here syndesmoses are, okay, slightly movable joints. So these are slightly movable joints, like we see between tibia and fibula, so an interosseous membrane. Okay, so these are also not filled with fluid, and synchondroses are also not fluid with, okay, fluid. So here it's the synovial joint, so that will have a fluid filled synovial cavity. So here the ball and socket will also come under, okay, this category. So it's okay, so I suggest to remove this option and, okay, fill it with another option, okay, like all of these. Okay, so here then the right option will be the synovial joints. So true joints are synovial joints. The next question, innominate is. So here we have two words for this. One is called innominate bone. So this innominate bone is the one that is, okay, part of pelvic girdle, and this innominate bone is formed by the fusion of three bones, that is ilium, then ischium, and then uh, pubic bones. So and this is the part of pelvic girdle. Okay, and again one more word we have in this case is the innominate artery, is innominate artery. So here we know from the left ventricle the dorsal aorta arises, and the dorsal aorta, okay, it takes a U-turn over the dorsal side, okay, and you will see here the branches arising from the aorta here, okay, right, and this is from the left ventricle, and it is carrying oxygenated blood to all parts of the body, right, and this one again will divide into, okay, two branches. So here this is what we call the brachiocephalic artery. So this is also called brachiocephalic trunk, artery or trunk, and this one has another name that's called the innominate artery. So it is called innominate artery. So this is the brachiocephalic artery or brachiocephalic trunk, and this brachiocephalic artery or trunk, it will divide into, okay, two branches, and one of the branch is called right common, it's called right common carotid artery, it's called common carotid artery, and one more we have here is the right subclavian artery, right subclavian artery, okay, right. So we have two words, the innominate, okay, bone, and innominate artery. So innominate is a nerve, no, it's an artery, yes, it's not a vein, and of course it's a part of a skeleton, as I told, it is a part of the pelvic girdle, so it's a part of skeleton and is also a artery. So that is also an artery, so hence the option D will be the right option in this case. The next question, human vertebra is. So here there are different types of vertebrae, right? Most of the mammals, okay, I mean all, almost all mammals will have acaelous vertebrae. So I'll just mention all mammals have acaelous vertebrae. Okay, so as the humans also belong to the group mammals, so they have acaelous vertebrae, but we will see what actually all these are procaelous, acaelous, and so here the coelus, which means a cavity, what we see in case of centrum. So in the last session, in the, I mean, previous sessions, we have seen the structure of a vertebra. So it has a body, which we call it a centrum, and if you find a, okay, depression on anterior end, so if you are seeing the vertebra from lateral side, so you may see something like this. Okay, so here, so is a region, so you can see here there is a cavity, this is a coelus, there is a cavity, so that's anterior region, and say this is the posterior. So the posterior region, you have a convex side, whereas the anterior region you have a concave surface. So have a concave surface. Okay, and here you see, so this is the spine, and again you will find another vertebra below it. We know they are present one above the other, and present like this. So this will be the neural canal through which actually the spinal cord passes through, and this is a neural spine. So this and this, these are neural spines. Okay, so you can see the cavity present on the anterior end and not on the posterior side. So such type of vertebrae, we call them procoelous. So procoelous type of vertebrae. Right, coming to amphicoelous, you will see the amphicoelous, as the name indicates, it will have concavity on both the ends. So you will see the concavity on both the ends. Right, so this is the neural spine. I'm telling that already you are seeing it from the lateral view, not from the top or the bottom. Okay, so here you may find, so here this is as usual the neural canal, and this is anterior side, and this is the posterior. So you can see the concave surface, so that is on both sides. So concavity on both sides, so then it is called amphicoelous. Amphicoelous. And coming to acoelous, you don't find any cavity here, so they are flat on both the ends. So you can see here they don't have any cavity. So you will see like this. Okay, so no concave surface, so on both sides it is flat. So here also, on this side also it is flat, so it is the, then it is called acoelous. Right, and I told that this acoelous you will see in almost all mammals. Then coming to heterocoelous, so in case of heterocoelous, the right surfaces are not perfect, so they don't show either concave or convex. Okay, so they are irregular, so you will see irregular like this. Okay, so in this case it's called heterocoelous, called heterocoelous. Right, so this heterocoelous type we may see in case of birds. This amphicoelous type of vertebrae you may see in case of fishes. So here you can see in case of fishes and some amphibians. And coming to this procoelous, in the case of this procoelous, you may see example in case of most reptiles. In case of reptiles, so most reptiles may show the presence of this type of vertebrae. So we understood procoelous and what is amphicoelous. It all depends on this the concavity, or whether it is present on one side, on both sides. So there are many other types, a few more types we have of this vertebrae, which here I am not discussing. So just out of these options we have seen, and now we understood that human vertebrae are acoelous, so they don't have the cavity on either side. So hence answer is A, C-less, that is C option is right. The next question, with respect to glucose metabolism during Cori cycle, the number of ATP the number of ATP formed by glycolysis and also the number of ATP used in gluconeogenesis respectively are. So for this we need to see about the Cori cycle. So this Cori cycle is also called lactic acid cycle, also called lactic acid cycle, and it is named after two persons who discovered this, Carl Ferdinand Cori and Jetty Cori are the two persons. Okay, so here what happens is during anaerobic glycolysis. So anaerobic glycolysis. So in the absence of oxygen, we know that the glucose will finally results in the formation of lactic acid that results in the fatigueness. So this lactic acid that is formed is transported to liver where it gets converted to glucose, a process what we call gluconeogenesis. Let's see how this process actually takes place. Uh, the Cori cycle. So here glucose, we know it will get converted to two molecules of pyruvic acid, and during this process it results in the generation of two ATPs. So that is ADP plus PI will result in the formation of two ATPs. Okay, so two ADPs and two PIs will result in the formation of ATPs, two ATPs. Now this pyruvic acid will get finally converted to two molecules of lactic acid. So here this entire process takes place in muscle cell. So this all process takes place in muscle cell. Now this lactic acid is transported from the muscle cell to liver cell. So now let's see in liver cell or hepatocyte. So here in the liver cell, so these two molecules of lactic acid will get converted back to two molecules of pyruvic acid, and this will finally get converted to a molecule of glucose. And during this process it utilizes ATP molecules. So it uses six ATPs. So that will result in the formation of okay right six ATPs. So here six ATPs are used. So six ATPs are used to form a molecule of glucose. So now as the glucose is formed from a non-carbohydrate source, so here that's what we say, the formation of glucose from non-carbohydrate source is what we call gluconeogenesis. Is called gluconeogenesis. So this process is what we call the gluconeogenesis, which takes place in the liver cell. So this all process takes place in the liver cell. Okay, so now with this cycle, now we'll understand here. Okay, so here they ask the number of ATPs formed by glycolysis. So here we know there are two ATPs formed, okay, during the process of glycolysis, and number of ATPs utilized in gluconeogenesis. So those number of ATPs used in gluconeogenesis will be 6 ATPs. So the answer will be 2, okay, formed, and 2 used in gluconeogenesis. So 2 formed in glycolysis and 2 used in gluconeogenesis. So option D is right. Okay, with this we will end this objective from locomotion and movement. In the next session we shall see a new chapter, that is neural control and coordination.